Okay, thank you to the organizers. Hello everyone, I'm Shuqiao Zheng from the Chinese Academy of Sciences. And today I'm honored to share our recent work with you, the network curvature analysis for single-cell data. In this presentation, I will provide a brief overview of the background and the related work. Next, I will present our proposed models in detail. And finally, I will showcase some experimental results. Recent advancements in single-cell sequencing technology have played a crucial role in biological research. For example, single-cell sequencing data can be used to predict cell lineage trajectory, create a human cell atlas, and promote precision medicine, and more. Single-cell sequencing data has high dimensionality, so dimensionality reduction is crucial. In this field of biology, common dimensionality reduction methods include PCA, t-SNE, EE, UMAP, and more. However, dimensionality reduction comes with the drawback of losing geometric information. For instance, it can't preserve geodesic distances. Consider 2 points on the sphere. The geodesic distances between these 2 points is this curve, relatively close. When we project the sphere onto dimensional plane, the geodesic distances is distorted. These 2 points is be aparted. Additional dimensionality reduction can disrupt connectivity. When we use the t-SNE to reduce the dimensionality of conducted components A from the sphere onto a 2-dimensional plane, the connected A becomes disconnected into several parts. Curvature is one of the most crucial geometric information. Curvature characterize the degree of bending. As the cartoon shows, this is a positive curvature space. Saddle surface has negative curvature, and flat surface has zero curvature. It has different applications in various biological networks. We will show in the next slides. In brain networks, curvatures reflect information that other classic network metrics failed to uncover. In cancer network, curvature can distinguish cancer samples and healthy samples. In gene and metabolic network, curvature can reflect the spectrum of continuous with multimodal in gene and metabolic. Curvature. What does curvature reflect in single-cell networks, and how to estimate the curvature in single networks? We proposed the BioCurvature framework on single-cell sequencing data. The algorithm consists of 4 main steps. First, we approximate geodesic distances on the cell method. Then we compute the distribution for each cell, followed by calculating the Washington distance between distribution. Finally, we obtained the node curvature. Next, I will explain the details of each step. Step 1. We approximate geodesic distances between two cells by calculating the shortest paths in Kn graph. Step 2. We calculate the distribution for each cell, encoding neighboring node similarity. The probability distribution Pi for cell I is calculated by normalize the graph Laplacian matrix. After giving distribution to each cell, we next compute the Washington distance between cells. This distance is calculated by optimal transport and measures the distance between two distributions. With the Washington distance and geodesic distance, we calculate the Oliver-Ricci curvature. Intuitively, negative Oliver-Ricci curvature reflects a bottleneck in manifold with two parallel lengths tend to go disperse. Positive Oliver-Ricci curvature represents nodes with high similarity, which forms a clique. Finally, we obtain the node curvature by weighted averaging. Next, we show our experimental results of methods. We first simulate random points from sphere. We calculate the curvature from each point, and ideally, this curvature should be identical value. We found that our estimate value is very close to identical value, and meanwhile, the estimate value has very small variance, indicating our estimation is very accurate and useful. On another example, we also random 2000 points on the saddle surface, and we found that the diagonal curve with low curvatures, which is consistent with the theoretical results. It also indicates the value is meaningful. Next, we will show real data. First, we test the human pre-implantation data. We call the HPE data. This data is collected cells at multiple time points from day 3 to day 7. As you can see, the embedding structure of data is very complex. It's a tree structure with multiple branches. We calculate cell curvature. We calculate cell curvature and found very interesting results. The PPT is have something wrong. First we found that most of the low curvature cells are located at bottleneck regions, indicating that they are at transition states, bottleneck regions. Well, on the other hand, For the high curvature cells, most of them are located in high density regions, indicating that they are in the high density clusters. They form cliques which have similar behaviors. Our results can show that cell curvature can be an important indicator of the cell types. Now we test the we also test another time series data from mouse embryonic brain context development. This data can develop to multiple cell states from early progenitor states to differentiated states. RPs is homogeneous, and neurons has various types. We calculate the mean curvature for each cell type. Interesting, we found that in the early differential states, this cell tend to have higher curvature. While at the differentiated state, they have low curvatures. This indicates that curvature can quantify the diversity of differentiated neurons. Finally, we test our data on the blood sample, which is called PBMC samples. It's most studied samples. These cells contain many immune cells, including T cells, B cells, natural killer cells, and more. Interesting, we calculated the distribution of the curvature of each cell type. They are not uniform distribution, but multiple distribution, which means that T cells have multiple subtypes to respond to different immune response, which is consistent with the immune study. In summary, I think I have evidence to you that our curvature is very accurate and useful to explore the single-cell network to provide biological insight from this analysis. We also provide uniform codes available. If you are welcome to try to use it. I'm looking for your feedback. Thank you very much. Finally, I thank to my collaborators. Now I have very happy to take your questions.
Thank you, Shuqiao. So we are open to questions and comments. Please come to in front of the microphones. But I guess we have to do two-dimensional representation also outside of the single cell analysis. So I guess many are interested in this new representation method. Okay, please.
Hello? Okay. Hi. Thanks for the great and interesting presentation. I have a question about your definition of your curvature on the single cell dataset. So based on my understanding, please correct me if I'm wrong, the curvature is defined based on the shortest distances between the networks inferred on the single cell data. Is that correct?
Yes.
Okay. So my question here was, have you compared your curvature-based approach to a more standardized clique analysis? Because it seems to me mostly you're looking at particularly shaped cliques on the single cell network. So have you compared with the standard clique analysis? And if so, can you give some insights about it? Thank you.
Yes. Can we go back to the slide?
Can you make the slides back to the screen? I'm not sure where are the stuff are. Thanks.
curvature, the positive curvature means the click we according to the density path paper, they cluster the cells into 14 clusters. The positive curvature means the nodes have high similarity. They are mostly located in the center of the cluster, in the cluster, and in this part and this part means the differential, the early differentiated and the differentiated. They are the same cell types. So they are also have high curvatures, and this cluster has high curvatures.
Okay, thank you.
Thank you.
Thank you for a great talk. Can you please explain I'm not sure I understand the curvature. So if you go back a couple of slides to the example of your negative curvature, either the image with the peanut or the very big, a little bit further back. When you have either the peanut, this one is good. So in the blue area, right? You say that's negative curvature on the left side of the image. It looks like a peanut with red and blue in the middle, right? So that's actually a saddle area in which you have positive curvature on the x-axis and negative curvature on the y-axis. So a negative, right? Do you understand what I'm saying? So I'm not sure why you call that a negative curvature. Can you explain that? No, on the left. Yeah, in that area. So on the y-axis, on the x-axis it goes like this, so it holds water. On the y-axis it goes the other way around, so it doesn't hold water. So it's a saddle. The blue area is a saddle area, right?
Yeah.
So it has a positive curvature in one direction and negative curvature in the other direction. And I'm not sure why you call that a negative curvature. Negative curvature would be like maybe. So what do you do when you have different directions of the curvature in different direction of the space?
The negative curvature means over squashing.
Means what?
Sorry, my English is not good. If you are interested, we can later.
Yeah, go ahead.
I don't know if I can explain this. So this curvature is a curvature defined on networks, which approximates the curvature in continuous, geometry. And the Oliver-Ricci curvature is a curvature on the edge of a network because it's defined between node I and J. You see, the Oliver-Ricci curvature is defined between two nodes, which there is an edge connecting them, right? So the edge goes horizontally, it has a negative curvature. I don't know if I have explained this.
So you are saying that the Oliver-Ricci curvature is calculated for a specific edge in the network,
Yeah.
And therefore it's only on one direction, and it's either positive or negative, and in this case it would be negative for
Yes.
the blue area.
In this case, it would be negative because if you go horizontally, like the two parallel lines, right, it goes departed, right? So it should be parallel if it's on a plane, right? Just like the grid case there where the curvature is 0, right? There should be parallel lines. But on this manifold, right, it goes departed, right? It started from this very nearby point, right?
I don't want to take too much time. Okay.
Yeah,
We'll talk more in the break.
Okay,
Thank
sure.
you so much.
Okay, let's thank Shuqiao again.
Thank you very much.